or at least in the right order. I didn't forget half of them. So we're going to do chapter 14. So what this whole chapter focuses on is really primarily eukaryotic cells. What has to happen to take the pre-mRNA, which will be the initial transcription. So we covered how transcription works, okay? But now in eukaryotic cells, there's extra steps that have to be processed, or extra steps in processing that RNA to make it into a functional mRNA. And that's really what the focus of this chapter is on. So before we get started with that, we'll do our 2 questions. So I thought for fun, we'll just give you exactly the same question as last time. See if you can remember it, except now it's worth a few more points. To start, yeah, okay. Oh, and while I'm thinking about it, if you happen to have misplaced a debit card last Monday, you might want to come talk to me. Not all of you, don't come down here. All right, we'll go to 1 minute on this one. So what's the answer from last time? Oh, I forgot. Hang on, I gotta always do this first time. Here we go. All right. So you could just look up in your notes, see what the answer was. You got your point. All right, let's try it for now. The new one for today is this fly is homozygous for mutation in the Fru gene. What is the sexual orientation of this fly? Oh, sorry. I'm gonna have to give somebody that job just to come up here and extra. I need my car waxed.
Awesome. 2 points on the test, and I'll do it.
Yeah, we'll do. Yes. Now, whether you're looking at answer D or not, I don't know if this fly is married or not. So, we'll go to 1 minute. Or whether it's, well, I guess a long-term relationship for a fruit fly would be about 2 months. If you want to grow old together, you can do it. Except for the mutants that we're working with, they only go about 40 days. Okay. All right. Put in an answer. All right, we'll go to 1:15 on this. Okay. So what's the answer? E? Well, that's actually one of them. Okay. So it turns out, well, it's sort of, okay. So a little bit of history on this gene. When this gene was first identified, they thought they had actually found the gay gene for flies because the males were trying to mate with other males. Once they actually looked at the problem a little more carefully, what they found was because of this mutation, the flies couldn't really distinguish the sexes, and they were simply trying to mate with anything that moved in the vial. So probably like kind of remind you of most sorority or fraternities. Okay. So now that I have your interest, let's get down to the science. All right. We're once again going to be redefining genes, what we mean by a gene. So we made it into this transcription unit last time because we added on sequences for the promoter. We go back to really a basic, the most fundamental definition, the most rudimentary information we'd have to have is what are the DNA sequences that code for the amino acids that are ultimately going to form the polypeptide. We're going to expand out from that point. Well, the first part of this is to look at the organization of the genes and recognize that there's a very major differences between the structure of these genes in prokaryotes and in eukaryotes. So as we've talked about before, prior to the molecular cloning technology that really came about in the 1970s and 1980s, almost all molecular genetics, genetic studies, biochemical studies and such really involved bacteria and viruses. And again, Francis Crick was involved in some of these ideas. From what I've read, Crick was just a really smart person who didn't like working in the lab. He liked coming up with ideas, and he really wanted other people to test out those ideas in the lab. But he had a lot of really good, insightful ideas. He also had a few ideas. One experiment I don't take time to talk about anymore, but I think it was a good example of where the thought process was right when you actually did the experiment. The whole idea was wrong. So he proposed that there was going to be this linear relationship between the DNA sequence and the actual amino acid sequence in the proteins. So he argued that these genes and proteins would be collinear. So here we see this term again. What he really meant was, if we look at the DNA sequence, and this upper strand is the sense strand for this particular gene, and then if we look at the messenger RNA, as we've talked about before, here's just a very good example for you. If you compare the sense strand to this RNA, the mRNA sequence, and just substitute the T's and U's, you have the same sequence. And then that sequence, in turn, would dictate what the amino acids would be. So that the number of nucleotides in the gene would be proportional to the number of amino acids. He didn't quite know at that time, the code hadn't been cracked yet, he didn't quite know that it was going to be 3 RNA bases would be a codon, so that there'd be 3 ribonucleotides per amino acid. But for the purposes of our discussion, this is pretty much what you see in bacteria and in viruses. This collinearity, so that you can say, okay, if this protein is 100 amino acids, then the amino acid coding sequence that we're going to see in the DNA is going to be 300 nucleotides. And that all worked out really well until people started playing around with eukaryotic cells. And then it showed up that things were very different. And there were several lines of evidence that showed that. One of them I want to show you is something called R-looping. So you can visualize DNA molecules very nicely with the electron microscope. I've shown you several examples of that. And the resolution is actually at a high enough level that you can distinguish single-stranded DNA from double-stranded DNA based on the width of the track that you're seeing on those electron micrographs. So the experiment that they did was to say, okay, let's take the genomic DNA, which would be double-stranded, and we'll isolate the RNA, the messenger RNA, and we'll do one of these hybridization experiments, or they also were called annealing experiments. So what we're going to do first is we're going to denature the double-stranded DNA. So now we have, so the darker color is single-stranded genomic DNA, the lighter color is the messenger RNA. Let's combine those together, cool the system down, just like I showed you in that other slide, and let the complementary sequences find each other. Now in bacteria, what you'll see is the sequences of the RNA. So the RNA, even though it has uracil instead of thymine, it's still complementary, and you can still form an RNA-DNA duplex. What you expected to see was that this sequence of RNA would anneal perfectly with one of these strands without any gaps. But in fact, what they saw were these kind of loops. That is, you had the messenger RNA, and then It was double-stranded with part of the genomic DNA, but also part of the genomic DNA stayed single-stranded. It looped out. These were the so-called R loops. So the interpretation of that experiment was that there were sequences in the genomic DNA that were collinear with the RNA right up to this point. Then there were sequences in the genomic DNA that were not represented in the messenger RNA. And then we once again have sequences in the genomic DNA that's represented in the RNA. So this was one of several lines of evidence that really made it clear that in eukaryotic cells you have these interrupted genes where you have these so-called introns and exons. To show you a little more closely what you're seeing, so right here, so here's the double-stranded DNA, and then this loop that's being pointed to by the arrow, that's a loop of the single-stranded DNA. So, any questions? Okay. So that's where this idea of the introns and the exons came from. So the exons get their name because they are expressed sequences. So the exons are the DNA sequences. So first of all, be clear. Exons and introns are in the DNA, not in the RNA. The exons are the DNA sequences whose corresponding RNA is present in the final mature messenger RNA. The introns get their name from intervening sequences. They are the sequences in the genomic DNA that are not represented in the mature messenger RNA. Okay, so that's how the naming came about. All right. Any questions with me so far? Okay. Now, what you see in general, as you go from the lower eukaryotes, the fungi, trypanosomes, and such, and then come up through Drosophila and up into humans, up into mammals, including humans, what you do see is there's a general increase in the number of introns in a particular gene and in the size of those introns. So, for instance, yeast, Saccharomyces cerevisiae, it's a eukaryotic cell. It has around 6,000 or so genes, but only a handful of those genes actually have introns. So in yeast, the vast majority of the genes don't have introns. All right. So if I gave you a question on the exam that said junior scientist has found some new gene in yeast, and instead of seeing these loops, all she found were these nice, smooth, double-stranded structures, what would that tell you? Does that gene have introns or not? That would tell you there's no introns. It would be more bacterial-like in that sense. Okay, so now, as you move up sort of into Drosophila, yes, most of the genes now have introns, and those introns tend to be larger. These introns in yeast, when they're present, may be a few hundred nucleotides, very short. As you get up into vertebrates and up into especially mammals, what you tend to see is the exons are very small, maybe only a few hundred nucleotides, and the introns can be very, very large, maybe over 10,000 bases or so, and lots of them. The dystrophin protein, to my knowledge, this is still the largest known gene. Yes? Say this again. That information is going to be missing when everything's all processed out. So that's what we're going to be talking about. Those introns have to be cut away and discarded. And so we're looking at comparing the mature messenger RNA in the cytoplasm as it's being used for translation, comparing that to the DNA sequence. The RNA sequences that are collinear with some of the DNA sequences will be the exons, but there will be sequences in the genomic DNA that are not represented in the mature messenger RNA. Those are the introns. So showing you some of the complexity of this problem, and sort of what this processing is going to be about. So what we're going to show eventually is the cell initially makes a pre-mRNA that makes a transcript including all the exons and all the introns. Then all the introns have to be precisely spliced out and all the exons knitted together. So this dystrophin protein, this is the protein when it's mutated, when it's not functioning properly, is responsible for Duchenne's muscular dystrophy. It's a big protein. It's about 4,000 amino acids. So in comparison to other cellular proteins, this would be a very large protein. That also means there's 12,000 nucleotides that are needed to code for the amino acids. However, this initial pre-mRNA that the cell has to make first is over 2 million nucleotides in length. So less than 1% of all these nucleotides actually are protein coding sequences. And I don't think I have it up here, but there's upwards of 100 introns in this gene. And so the cell has to make this 2 million nucleotide pre-mRNA and then correctly remove about 100 of these about 100 of these introns to get a functional protein. If you're thinking ahead a little bit, you recognize that if these introns that fall within the sequence that codes for the amino acids, if they're even off by one base, that's the equivalent of a frameshift mutation. So a lot of human genetic diseases are in fact due to mutations that alter the splicing that happens with these introns. But we'll get into that in a little bit more. A little more manageable picture is the ovalbumin gene from chickens. And what you see here is this is more, these exons are probably fairly close to scale. So the darker color is the exon. So this is the DNA sequence. The lighter color region are the introns. Initially, the cell is going to make a pre-mRNA that's going to have all of this. I heard something, but I don't know what I heard. Okay? We ready now? Okay. So now we're going to make a pre-mRNA that's going to be this full length, and then the processing is going to take each of these little areas, the dark areas, the exons, and put them all together to give you the mature mRNA. So how we go from here to here is really the substance of this chapter. Okay. And along with this came the discovery of self-splicing RNA and a Nobel Prize. Okay. So there's 4 major classes of introns. The first, you see a certain pattern, group 1, DNA polymerase 1, RNA polymerase 1. All right. These are found in some of the ribosomal RNA. This is where the self-splicing catalytic RNA was found. I'll tell you more about that a little bit later. Group 2 are found, these are some protein coding genes. These are within the mitochondria, in the chloroplast, and actually are being found in a few bacteria now. It was, for many years it was dogma. There are no introns in bacterial genes, but now we're finding some examples as more and more bacterial genomes are getting sequenced. These are also self-splicing. They use a different biochemical mechanism, and we're not going to go into this in any detail. The main one we're going to focus on are the nuclear pre-mRNAs. So these are the processing that's occurring with the transcripts made in the nucleus, in eukaryotic cells. They require a large complex called the spliceosome. We talked a little bit about the RNAs and the SNRPs and such. That's where, so the spliceosome is a ribosome-size organelle, really, but it's in the nucleus. And it's going to be responsible for processing the transcripts made by RNA polymerase II, the ones that end up coding for proteins. There's a 4th type of intron, we'll just note it here, that some of the tRNAs, not all of them, but some of the transfer RNAs do have some small introns. Okay, so now we'll just sort of redefine what we mean by this gene, reinforcing the point that just saying, oh, if we just talk about the DNA sequences that code for the amino acids, that's not sufficient. So this just brings us back and refreshes your memory on what we talked about as a transcription unit, the promoter, the actual RNA coding sequences, and the terminator. The important point here is now we're adding in that this RNA coding sequence is going to include all of the introns and all of the exons. They're going to get made into one RNA molecule first, then the introns will be removed later. And also keep in mind that gene products don't have to be proteins. The vast majority are, but tRNAs, ribosomal RNAs, these little small nuclear RNAs, the microRNAs and such, they are the final gene product. But most are going to be for proteins. Yes?
How would you, just going back a second, how would introns get into tRNA?
Oh, they're actually, I think usually they tend to show up in the anticodon loop. They're part of they're part of the initial transcript of the RNA, and it's a small intron that has to be processed out. Why it's there? Is there some selective advantage to it? It's not that I don't know if anybody's figured that one out yet. Clearly, one of the challenges of doing research in biology is biological systems don't always act in a way you think is logical, and they do things in what appear to be. Okay, so the question would be, why would there be natural selection for such a complex, probably error-prone process as these introns? And the answer is, when it now looks like we can make variants or isoforms of different polypeptides from the same gene, it now means one gene can code for multiple products. And initially when these introns were found, it was thought, oh, these are going to be the rare exception. Now they're clearly in the majority. Why the tRNAs have it? Good question. They're not that big to begin with. They're only about 75 nucleotides. Okay. So part of the puzzle was that investigators knew that in eukaryotic cells, the DNA was in the nucleus. They could find that. That was easy to find. They knew proteins were made in the cytoplasm. So it was Francis Crick who suggested that there would be some sort of messenger. Something had to shuttle the information from the nucleus out into the cytoplasm. The problem is, as you probably already know from general biology, in bacteria where they were doing these experiments, the mRNA turns over very fast. So the mRNA was very unstable, and so they hadn't really seen it yet. So they didn't have good evidence for this messenger RNA. So Crick was really hypothesizing that it must exist, and then people started looking for it. And what they did is there was a number of experiments that were done that conclusively showed the presence of this messenger RNA. One of the clearest examples came from experiments using bacteriophage. Since the DNA sequence of the genes in the bacteriophage are quite different than the bacteria, what they're finding is once they infected a cell with the bacteriophage, there was this short-lived RNA molecule that had an RNA sequence that was very different from what was in the bacterial cells, and therefore probably was the messenger RNA that was being used to make the bacteriophage protein. So that was just one of a lot of experiments that really identified this fairly unstable molecule. So it was hard to find at first because it was so unstable. They also noticed that this new RNA became associated with ribosomes. They knew the ribosomes were the site of synthesis for the polypeptide. So all this information started coming together and clearly laid out the existence of this so-called messenger RNA or mRNA. Okay. So any questions about that? Now we're going to look at this in more detail, as we tend to do. There's going to be, whether you're looking at a prokaryotic, actually the mature eukaryotic mRNA is actually very similar in structure to the prokaryotic. It's the pre-mRNA before all the processing happens where things are different. So if we look at them just very generally, there's going to be really three primary regions on the mRNA from either a prokaryote or from a eukaryote. At the 5' end, we have the so-called 5' UTR. This is for the untranslated region. What does that mean? What's translation? Well, the translation is that actual process where the ribosomes are reading the codons on the messenger RNA, putting the amino acids together, and making the polypeptide. So this is the part of the messenger RNA on the 5' end. So it's upstream of where the amino acid coding sequence is that doesn't get translated. So it's the 5' untranslated region. So it lies, just like the promoter is upstream of where transcription begins. This is at the 5' end. Sometimes people call it the leader sequence. And the key is it does not code for amino acids. In bacteria, just in bacteria, there's a sequence called the Shannon-Dalgarno sequence, named after scientists who found it. And what that sequence does is that helps the ribosomes know where to attach to the messenger RNA. So this is a ribosome binding site. So this will be important for translation. So you can imagine that mutations in this sequence would probably do what? If the ribosomes can't, if that sequence is changed, the ribosomes don't interact properly, you're not going to make any proteins. Okay. You had a question?
Yeah, would the AUG codon be part of the untranslated region?
No, the ATG is, we'll get that to in a minute, that's the start codon that starts the amino acid coding sequence. So this is the sequence that, if you know about the ATG start codon, this is the sequence that precedes it, upstream of the ATG start codon. Okay. Then in the middle is the protein coding region. This has often been called the open reading frame, because in many cases, if you didn't actually know what the protein coded for, it was a sequence that had a start codon. So we'll talk about this in more detail when we get to translation, but all prokaryotic and eukaryotic genes, with only a few exceptions, all start with an AUG start codon. So they all start with methionine, because for methionine, this is the only codon that codes for it. So this open reading frame starts with the AUG start codon, and basically was the reason they said open reading frame is now just follow that RNA sequence codon by codon until you come to one of the 3 stop codons. So the open reading frame is defined as the start codon at the 5' end and a stop codon at the 3' end. That's the actual sequence that the ribosomes will read, bring in the tRNAs, and make the polypeptide. And then downstream of that stop codon is simply the 3' UTR, the 3' untranslated region, because that sequence is not translated. It doesn't code for any amino acids, and it really seems to be involved more with the stability of the mRNA than anything else. So the most important sequences are on the 5' UTR and the actual protein coding sequences. So if you want to see it sort of in a cartoon form, and this generally would work, you would see the same general structure for both prokaryotic and eukaryotic mRNAs. So here's the 5' end. Here's the 5' untranslated region. Part of that is the Shannon-Dalgarno sequence in bacteria. This is where the ribosomes interact with the messenger RNA, so that they're now positioned to start translation with a start codon here. Then we go through the open reading frame or protein coding region, all the way down here to the stop codon. That's the end of the protein coding region. And then you have the 3' untranslated region, or the 3' UTR. All right. Okay. So let's take that and give it a break for a minute and see what you can take away from that. Oh, this is another vampire bat. Oh, okay. We'll go to 1 minute on this one. Okay, get your answer in. Put something in. All right. So, for those of you who can remember back 3 minutes, what's the answer? You guys are way too young for the short-term memory loss problem. Okay. So now we get to the fun stuff. This pre-mRNA processing. So this is going to be in eukaryotic cells. All right. What was the key point of it? Okay. So the key point of this slide is in bacteria, when the messenger RNA is being made by transcription, it doesn't need additional modifications. It's ready for translation. So, and since protein synthesis and transcription occur in the same place in the cell, you literally have the ribosomes attaching to the messenger RNA and beginning to make the polypeptide before transcription is even completed. You can't do that, okay? And there's going to be a mechanism of gene regulation called attenuation, which ties together. It's actually it'll be part of an extra credit question we'll get into later on, but it involves the interaction between the process of transcription and translation occurring simultaneously. Could you have that kind of a regulatory mechanism in a eukaryotic cell? If the transcription is occurring in the nucleus and the protein synthesis in the cytoplasm, they're physically separated. So this process of attenuation only could work in a bacterial cell. The other is, as you'll see, there's a lot of extra biochemistry going on to this pre-mRNA before it's ready to be sent to the cytoplasm. Okay, so that's what all that's really about. All right. So there's really going to be 3 main modifications. We're going to have this 5' cap structure I'll explain to you in a minute. So this is going to be a special structure put on the 5' end of the messenger RNA. Then there's going to be a process called polyadenylation, which is a series of A residues that are going to be added to the 3' end. And then we have to cut out all the introns, get all that spliced together. Then transport the messenger RNA out into the cytoplasm, and then it can be used for protein synthesis. Okay. So this 5' cap structure, this is going to be added after transcription begins. All right. It's a special guanine nucleotide, so it's a G. So sometimes this has been called the 7-methyl G cap because there's several unique things about it. First, there's methyl group, CH3 group. Okay, so first we're going to modify this guanine residue. I'll show you that. Then on the next base, there'll be a methyl group added to the 2' hydroxyl of the ribose. And sometimes even on the second one. I'll show you this in a diagram. This cap structure is required for 2 really important reasons. One, it stabilizes the messenger RNA against attack by exonucleases from the 5' end, and it's absolutely required for translation. So if you don't, without that cap structure, the messenger RNA it doesn't interact properly with the ribosomes, and it cannot be used for protein synthesis. So it's a critical structure. Actually, I did my master's degree with some of the scientists who actually did those experiments showing that how important this cap structure is. All right. Here's some unusual features about it. First of all, we keep talking about this phosphodiester bond is 3' to 5'. With this cap structure, this guanine nucleotide we're going to add, it's going to have a unique 5' to 5' linkage. So this is not your standard 3' to 5' to 3' linkage. There's going to be 3 phosphates instead of just 1. And there's going to be a methyl group that's added to position number 7 on the guanine base. And then on the next base, as I said, there's going to be a methyl group added to the 2' hydroxyl. So here it is in a diagram. So the important point is this is actually the first base. So this would be the base that corresponds to +1 on the sense strand that we talked about before. Then, after transcription begins, but before it's finished, but let's say it's 100 nucleotides downstream, this modified G residue gets added. Right here, at position 7, this methyl group is added. And then what you see here is this is the 5' carbon. Remember, the phosphates are attached to the 5' carbon. So this is a 5' to 5' linkage, and all 3 phosphates are present, not just 1. And the idea is that this is really protecting the end of that messenger RNA from attack by the exonucleases that are in the cell, and this structure is required for proper interaction with the ribosomes, and therefore if it's not there, then this messenger RNA can't be used for translation. All right? So that's the 7-methyl-G cap, or the 5' cap. It's all referring to the same thing. The other kind of unexpected result is that the 3' end of the messenger RNAs are polyadenylated. What that means is there's a series of A residues, adenines, added at the 3' end. Depending on the messenger RNA, depending on the species, it ranges in length from about 50 to 250 residues of just A's, nothing else. The other important thing is there's not a corresponding series of T's in the DNA. These A's are added by an enzyme called the polyadenylate polymerase. So after the transcription is done, then these A's are just added in a template-independent enzymatic way. Okay. So why? Any guess why the? Or a lot of times it's just reading. So why would this? This could be another kind of added level of complexity. Any ideas on the purpose of this long poly-A tail? It helps stabilize. It's also known. It helps also with the translation process now. It does interact with some proteins that interacts with the cap structure and helps the ribosomes interact properly with the messenger RNA. Now, this poly-A structure is only on messenger RNA. It's not on ribosomal RNAs. It's not on tRNAs. Just the messenger RNA. So for a long time, molecular biologists exploited this and used that poly-A tail as a way to purify messenger RNA from all the other RNAs in the cell. With PCR now being as sensitive it is, you don't really have to do that. Okay. So putting this all together, what we have is, so here's our gene, our transcription unit. We have transcription. This is the pre-mRNA, so it starts at that +1 transcriptional start site we talked about. And it transcribes all the way through exons and introns. Okay, this shows it a little better. Last time, right towards the end, I talked about termination, and I said there's a sequence that tells the clipping enzyme where to cut the messenger RNA so that you don't really have defined termination sites. Well, that's what this is. So this consensus sequence is for the clipping enzyme. And so when it reads this sequence, then a short distance, about 11 to 30 nucleotides downstream from there, it cleaves it. So it doesn't really matter where termination begins because all of this is going to get removed. And so from this point right here to this part, what removes that? I know it's all the way back in that other. Remember the RAT1 endonuclease? All right. Thought you might remember it because I like the name of it, I think. Okay. So what you're going to have now, so then you're going to get the 5' caps over here, and this poly A tail gets added on to the 3' end enzymatically. So there is no template, there are no sequences in the DNA. It's just an enzyme reaction. It just adds sequentially A residues to the 3' end of the mRNA. So the mRNA is modified on both ends. You got the cap structure on the 5' end, these poly A tails on the 3' end. Okay. Interesting, nearly all eukaryotic mRNAs have been looked at have the poly A tail. One notable exception are the histones. So histone mRNAs lack the poly A tail. Why might that be? Is stability important for the histone mRNAs? Do they need to be around for a long time? When are they made? When would cells express the histone? When do they need to make the histone proteins? Just during S phase, during DNA replication. So they are a very short-lived messenger RNA. So they are unique. As I said, the vast majority, if you're going to have to bet your lunch money, if some new gene got isolated, is its mRNA polyadenylated in the eukaryotic cell? The answer is yes. The histones are one of the very notable exceptions that they are not polyadenylated. Okay. So we've done 2 of the 3 things that we need before this messenger RNA is actually ready to be used for or at least transport to the cytoplasm. The third and the more complicated process is this removing of the introns. All right. So in most cases, the capping is finished, the polyadenylation is finished. Then you have the splicing. This is all happening in the nucleus. All right. So I think this is where I started. Yes. Yeah. So you should have just known everything I just told you, and we could have just started here. Okay. As usual, more consensus sequences. What we're really having to do is I'll show you in detail, you're going to have the junction between where the exon ends and the intron begins. At that 5' end, and I'll show you in a figure, there's this GU consensus sequence. Actually, this is pretty much invariant. You almost have to have, if these bases get mutated, then the splicing doesn't happen. So this is more like invariant than being a consensus sequence. And so this G is going to be really important in the mechanism of how this intron is going to get removed. Then at the 3' end, you have this AG invariant sequence. And then what doesn't make sense, but this is, I'm just reporting the news, is there's something called the branch point. So there's an A, so here's the consensus sequence, basically lots of pyrimidines, a purine, but the important one is this A, which is about 18 to 40 nucleotides upstream of the 3' end. This is going to be important for the mechanism of removing the intron. So as it shows you here, if you mutate that GU at the 5' end, the AG at the 3' end, or the A at the branch point, any of those sequences mutated, you're going to block the splicing of that intron. Yes.
What would happen if it doesn't get spliced?
Okay. So the question is, what happens if that intron doesn't get spliced out? What does it really become? It's now an exon, right? It's going to be in the messenger RNA. So let's say it's sitting in the middle of an open reading frame, and you're going along and reading each of your amino acids, and then you got all this extra sequences gets thrown in. It's like an insertion mutation. So it would, especially if this was in the protein coding sequence, you'd get a completely non-functional protein. As I said, a lot of human genetic disorders are mutations that affect the splicing rather than the traditional single nucleotide change that changes an amino acid or something like that. This is much more prevalent. Okay. So here's the important points. So the lighter color region starts the intron. So what we really want to do is we want to join this G that's on the 3' end of exon 1 to this G that's at the 5' end of exon 2. That's the end result. And throw away the RNA that's in the middle. So here is that GU sequence. Okay, so this G is the 5' end of the intron. This G is the 3' end of the intron, and there's the branch point. Those are the three sequences we have to look at. Okay. While there is self-splicing RNA, these RNAs need help. So this is done with this structure in the nucleus called the spliceosome. It's what's going to be responsible for carrying out these splicing reactions. So I think of it more on the scale of like a ribosome. It is an RNA protein complex. So it has the small nuclear RNAs that we talked about before. So they're short in the 100, 200 nucleotide range. But those are, it's an RNA protein complex. So this is what we call these snRNPs for small ribonucleoprotein particles, or we just call them SNRs. All right. So the splicing reaction, it wasn't exactly what people thought it would be. And it's been called a lariat model because when the intron is removed, it's going to look like one of these lariats that American cowboys had. So I found this cartoon this morning. In case you haven't been to a dude ranch or you don't, I don't know how many roped cattle on the weekend or something like that. Okay. So that's how it got its name, this lariat model. So first, we're going to have to cut the pre-mRNA at the 5' end of the splice site. So that's going to leave that G that I was pointing to. That's going to be free to interact with the A. That's where it's going. Okay? Now, if you think about this a little more carefully, the phosphodiester bond between the 3' nucleotide of the exon and that G5' base, which is the first one of the intron, you could cleave that on either side of the phosphate. Okay? We need to cleave it on the side that leaves the phosphate associated with that G, because it's going to have to form a phosphodiester bond. If you kind of diagram that out, look it up, you can see that when you break these bonds between these adjacent nucleotides, you can break it on either side of the phosphate. In this case, it's got to be on the side of the phosphate that leaves the phosphate attached to the 5' carbon of the G that is the first base of the intron. Because what's going to happen is that 5' end is going to come around and interact with the A residue. That's that branch point we talked about. That's why it gives you this structure that looks like a lariat if you look at it under the electron micrograph. What it predicted, and nobody believed until it was shown, is it has to make a novel 5' to 2' phosphodiester bond. So that A at the branch point actually has 3 phosphodiester bonds. The 2 normal ones, you'd have one at the 5' carbon, right? One at the 3' carbon, and now you have a third one on the 2' carbon. So when these investigators were trying to publish this paper, they had to show very rigorously that in fact there were 3 phosphodiester bonds associated with this A residue at the branch point. Okay. The second step is to now cleave the intron down at the 3' splice site. And now simultaneously you have to bring the 3' end of exon 1 to the 5' end of exon 2. And then the intron gets released in this lariat-looking molecule. And people have actually visualized that under the electron micrograph. and then it breaks down and it all gets degraded. And so eventually the intron, the sequences in the RNA that correspond to the introns are now completely degraded, and the two exons are joined together. So this is in a diagrammatic form. So here's exon 1, the intron, exon 2. So we cleave right here at the 5' splice site, and here's that lariat structure coming around. If you look a little more closely, this G right here is the G that was at the 5' end of the intron. But now it's formed a phosphodiester bond with the 2' hydroxyl of the A residue. So it loops around. The two exons get joined together. This lariat-shaped intermediate for the intron gets degraded, and the RNAs get and the ribonucleotides get recycled. Okay, and that's just one of them. So in the dystrophin, the cell has to do this with about 100 of the introns. Can you also see where if you make a single base mistake, and if this is part of the open reading frame, it's the equivalent of a frame shift mutation. So it has to be done very precisely. Also, in some cases, there's some mutations that have occurred, let's say, let's say there's a mutation that occurs, about halfway between the 5' end of the intron and that branch point. Would that probably affect the functionality of the gene since you're throwing these sequences away? In most cases, no. There are some known examples, though, that end up creating a new splice site. The mutation creates a new splice site and causes the cell to splice in the wrong place. So there's a number of ways that this thing can go wrong. Okay. So as I said, all of this is being hammered out on these spliceosomes. And the key to this, and we're not going to go into it in any detail, is these small nuclear RNAs that are part of the spliceosome are complementary to the sequences on the ends of those introns. The question is, once I cut the introns on the two ends, my exons float away. How do I hold them together? They're being held in place on the spliceosome because the RNA molecules are complementary to the sequences at those junctions, so they're holding the RNA in the right place while the intron gets snipped away and the two exons get joined together. Okay. There's a whole range of these small nuclear RNAs, and I will spare you of going through all of them. The main take-home message is these spliceosomes are an RNA protein complex. One of the main functions of the RNAs in the spliceosome is to hold the pre-mRNA in the right place so that these reactions can take place and the exons can be joined together properly. Okay? One other comment is just on the organization of the nucleus. I've mentioned this before. For many years, it was thought that the cytoplasm and probably the nucleus too was just basically this organic soup. Molecules just diffuse. And then people started looking at the rates of some of these reactions and realized that the reactions are going faster than diffusion would allow them to go, and that there has to be an organization, and there actually is. There's a cytoskeletal structure in the cytoplasm, basically a cable car system with little molecular motors that are moving molecules back and forth. Also, you have a similar organization in the nucleus. So these spliceosomes are not just randomly distributed throughout the nucleus, but they have actually very specific locations. They do this, it's really very kind of cool. Are cells three-dimensional or two-dimensional? They're two-dimensional in your textbook, but they're actually three-dimensional in the real world. So you can do, now you can do it with basically confocal microscopy, but you literally do electron micrographs slicing layer by layer by layer through the nucleus, and then reconstruct all of those into three-dimensional figures, and then you begin to get some evidence about this organization. So here the key point is there's a very specific organization to the nucleus, and these functions take place in discrete locations. Okay. What came out of this, and I was talking to one of the students about this, so I'll apologize because I already told him the story. So what came out of this was the discovery of self-splicing RNAs. So this was Tom Check's lab. He was at Boulder, Colorado, I believe, at the time. And I heard him give a talk, so I'm pretty sure that this is the way the story really works out. And this happens with so many discoveries in biology. They were just asking a very straightforward question. They wanted to find the proteins that would catalyze these splicing reactions. They wanted a simple model to work with, so they had a small little RNA that had a small intron in it, and they could very easily assay for when that intron was removed. But as they were doing these experiments, they kept seeing that their control would also splice. So what would be, in this experiment, what would the control be? If you're trying to find the proteins that catalyze this reaction, your control would be the RNA without any added proteins. So you got this new wet behind the ears grad student who can't get this experiment to work right because he's a screw up. Then he's contaminating the control. That's where you start. Okay, you're a screw up. I'm going to give this to somebody else. So you give this to your more senior grad student. They do the experiment and get the same result. So then you bring in the postdoc and you have them do it, and they do the same thing. You've done it 3 times now, 3 different people, and you get the same unexpected result. So you have to kind of open your mind at that point and go, what if this is correct? What if these RNA molecules are actually carrying out this reaction on their own without any added enzymes? So then they rigorously proved that in the absence of any possible protein, these RNAs could catalyze the removal of this intron. And this was the discovery of self-splicing RNAs, molecules we call ribozymes. He won the Nobel Prize for it. And yet it wasn't a case of reading the literature and predicting the existence of self-splicing RNAs. They just kind of stumbled onto it. Okay? And, if you want to work on the Nobel Prize, I mean, the 3 scientists who shared it this year, I believe it was close to $1 million for the prize. It depends on market. So only be creative in the years when the market's doing well, because it's really the gain on the Nobel Trust is what you get. Would have been my luck. I would have probably won it in 2008 or something, and I probably would have owed them money. Yeah. Oh, look, you got $12.32. Okay. So we talked about those 4 introns. Well, the group 1, the group 2 are the self-splicing ones. Okay, so means these RNAs have true catalytic activity. Just to make it, so these group 1s, these are not, it's important, these are not these nuclear pre-mRNAs that code for the proteins. That's not what we're talking about here. So these are some of the small little ribosomal RNA genes. They tend to be in the mitochondria and such. As I said, the group 2 are also in the mitochondria. The mechanism is a little different. We won't go into that. What you're really seeing here is the role, and this comes back to an important point, it's the structure of the RNA that gives it its enzymatic activity, or its biological activity, whatever it's doing in the cell. So it's those inverted repeats and such that give it a 3-dimensional structure, very much like proteins. And again, these catalytic RNAs, and you can buy some of these commercially now, have been given the name ribozyme for the fact they are riboenzymes. Okay. So it comes to this question like, why? Why are what I said at the beginning? Why would these interrupted genes, we're finding a few cases in bacteria now, but they're not very common. But they're very, very common in eukaryotic cells, especially in mammals. We've already talked about how they're complex reactions, they're very error prone, lots of opportunities for mistakes. Why would there be a selection for this? And it didn't really make sense until we realized that the cells don't always just splice the exons 1 to 2 to 3 to 4 in just one way. But rather, they can do differential splicing. They can put the exons together in different combinations and make isoforms or variants of that particular protein. This helps explain why the human genome only has about 20 to 25,000 genes. Since it looks like maybe at least 60%, maybe even more, 70, 80% of those genes have this differential splicing. We'll talk about more of that later on. You can make different variants from the same polypeptide. So the two genes that we work on in Drosophila, MSRA and MSRB, they're just two structural genes, but there's about 6 to 8 isoforms for each one. So between those two genes, the cell can make about 12, 14, maybe 16 different polypeptides from the same gene. So this is why a human cell might be able to make 100,000, 150,000 different polypeptides from only about 25,000 genes. So you're just getting different polypeptides derived from the same gene. So this is what it wants to, they call it alternative splicing. A lot of times you call this, you'll see this as differential splicing. It's the same thing. So it's a very simple gene. We got 3 exons, exons 1, 2, 3. Just like we talked about before, here's the pre-mRNA. You make the whole pre-mRNA. The 3' cleavage site is going to be right here at the end of exon 3. So you add the poly A tail on here. And now you come to the splicing. You can splice it the way you would think it would happen. And you remove introns 1 and 2. And you splice exon 1, 2, and 3 together. And you get some polypeptide out of that. Or the cell can skip from exon 1 to exon 3, treat exon 2 as though it's part of the intron, and get rid of that sequence. And now you've made an isoform or a variant of it. Okay? What you don't see is you don't see the cells going, oh, let's go exons 1, 3, and 2. You don't change the order. You just change which ones you skip over in the way you splice those together. But if you start to have 100 different exons, you can put an incredible range of polypeptides together by splicing them in all different combinations. Yes?
So this is literally how a gene can code for multiple traits in the organism?
No, I wouldn't say for could be. So keep in mind, these proteins, they're going to be related to each other. Okay? But for instance, in the ones that we work with that I was telling you about, we have about 6 isoforms for MSRA. What the data looks like is 2 of those are spliced in a way that targets them to go to the mitochondria. The other 4 function in the cytoplasm. So where in a eukaryotic cell, where that protein is functioning can be as important as what that protein is. So you could easily say, okay, these variants, these are going to the endoplasmic reticulum. These are going to get secreted. These are going into the nucleus. These are going into the mitochondria. So they're related to each other. But yeah, depending on what the function is. Okay. Another alternative that you can see, it's not as common, is just change where the mRNA gets clipped on the 3' end. So you can have multiple, instead of having just one clearly defined cleavage site that you clip off the messenger RNA, some messenger RNAs have multiple 3' cleaving sites. So you can see this here. So the same thing we had before, but what it's indicating with these numbers, 1 and 2, are 2 different places where the molecule can be cleaved. If it cleaves at number 1, then we're just going to get exons. We're going to get exon 1 and a part of exon 2. If we cleave at site number 2, we're going to get more of exon 2. Okay. Now later on, at the beginning of near the beginning of the course, I kind of joked about a gene called sex lethal, SXL. I thought the name was funny and such. There's going to be something very similar to this going on. What can happen is you can have a protein that binds to the mRNA and covers up site number 1 and forces the system to cut at site number 2. So if the cell makes that protein, then you get this variant. But if this is the preferred site and the cell doesn't make the protein, then you cut it here. So you can control the outcome based on whether or not you make the protein that masks that site. So it gets into these finer and finer levels of gene regulation. But as I always promise, we'll get into more detail later on. Okay? All right. So yeah, just a little bit about the importance of these so-called isoforms or variants. I got a qualifier on here. You got to be careful. Pretty uniformly, variants is used as a term to talk about the different mRNAs that come out from differential splicing from the same gene. Just like everything we've been talking about. Isoforms are also used in the same context. But as we talked about before, eukaryotic cells have families of genes like the beta globin genes. Sometimes the term isoforms is used to talk about closely related proteins that are coming from different but closely related genes. So it serves double duty here. If you want to be really safe, you can call them variants. If you really want to make sure people understand you're talking about differential splicing within the same transcription unit, then use the term variants. But isoforms is also correct, but it's got this dual usage. This number, I don't know how accurate this is, but at least well over half, probably even 3/4 of human genes. In other words, when this was first found, it was thought to be the rarity. Now it's clearly in the majority. And as I said before, a lot of genetic diseases are mutations that affect this splicing. In fact, 15% of the single base mutations, the traditional ones they tell you about in general biology, actually are in fact result in splicing errors. As I said, some of these create new splice sites, so they are in places that shouldn't give a problem, but in fact end up changing this. They make a new splice site that's even stronger in attracting the spliceosome mechanism than the original site. All right. Since you need a break from me, here we go. All right. Let's see this one. Looks like we should be able to go to 1 minute on this one. Okay. Place your bets. The window's closing. All right. So what's the answer? All right. See, all you got to do is be able to think back 3 minutes ago. You can answer these questions. All right. So how many of you think that's a fairly complicated process? Oh, we'll admit. All right. You think that's crazy? Here's another one. There's something called RNA editing. It's a limited number of examples, but it was found way back in the 80s, so that's all history for you guys, so it's all fair game. the important point here is this is a process that's going to change the mRNA sequence after it's been transcribed. So you're going to have nucleotides in the RNA that are not coded for in the DNA. So this is called RNA editing. And it really involves inserting and deleting some bases. But again, this is after the RNA has already been synthesized. And there's really two mechanisms here. One was the discovery of so-called guide RNA. So here's the mRNA before it's edited. So this would be would these sequences correspond to sequences of the DNA in this top figure? Yes, that's as it was transcribed. Then these guide RNAs have homology. They're complementary to this sequence, and they can form this complementary pairing. And through a mechanism that's not really well understood, but at the level at which it's understood is beyond really the scope of this course, the end result is they end up inserting bases into the final mature messenger RNA. So this is what the pre-edited mRNA was, but at the end, all these extra U's have been added. What seems to be the most common purpose for this is there's been mutations in the DNA sequence that have caused a large number of frameshift mutations, which would really make the gene non-functional. This actually restores functionality by removing those frameshift mutations. So this is RNA editing. As one mechanism, it's changing the RNA sequence after the fact, after the RNA has already been synthesized. So these guide RNAs are one mechanism. Another is to do this biochemically. If you look at the structure of cytosine, you'll see that you can convert it to uracil by a deamination reaction. That is, to remove the amine group. So when we get into the chapter on DNA repair, this is one of the, since these are somewhat unstable linkages, this type of deamination can occur in the DNA, and now you've got uracil in the DNA, so there's got to be a mechanism that can repair it. Here, this is being exploited to be able to change the sequence from a C to a U. And for instance, that could then alter a codon to become a stop codon and such. So you can either change the sequences through these guide RNAs or by biochemically changing the bases. In this case, as an example, a deamination of cytosine to yield uracil. Okay. All right. tRNAs, what do we want to say about that? So the tRNAs are an adapter molecule. They are going to be the molecule that brings the amino acid. So they have an amino acid at one end, and then they have the anticodon. So they're really the molecule that's going to bring the amino acid to the ribosomes during protein synthesis. There are 20 amino acids found in biological systems and proteins. Are there more amino acids in the world than 20? Yeah, a lot of them. There's 20 that are used in proteins. However, most organisms have 30 or 40 tRNAs that are each encoded by a different gene. Why would they need more tRNAs than they have amino acids? Well, what is the tRNA doing? You have the anticodon has to match the codon. Is there only one codon for each amino acid? No. In most cases, there's only two amino acids that have only one codon. Methionine, I think tryptophan. So what you have are more tRNAs than you have amino acids because you have more codons than you have amino acids. When we get to translation, we'll talk about that in more detail. But I want you to notice here, and you don't have to know this in a biochemical extent, But tRNAs not only have the usual nucleotides, they have a number of fairly complex modified bases. Pseudouridine, ribothymidine, and such. So I'm not going to ask any specific questions about the details of that, but recognize that tRNAs especially have these chemically modified bases, and they're important for their activity. Okay. The other thing is cells have to make a lot of tRNAs. So cells normally have multiple copies of the genes encoding each of these tRNAs. In eukaryotes, these tRNAs are made by RNA polymerase III. And there's also a lot of cleavage and modifications and such before you get to the actual functional molecule, but we're not going to go into any of that detail. And just make a note that some tRNAs have a small intron in them. That's actually pretty rare. Any questions? All right. Ribosomal RNA, critically important. Cells have to have a lot of it. As I've said before, if you isolate total RNA from cells, probably, I've got 90% here, it's probably more than that. Probably 95 to 98% of all the RNA in the cell is going to be ribosomal RNA. Cells have to make a lot of it because they've got tens of thousands of ribosomes per cell. All right. In bacteria, these genes for the ribosomal RNAs are dispersed throughout the whole genome. But in eukaryotes, there's an organization. And this is very conserved through all eukaryotes. So there's 4 major ribosomal RNAs. So each half of the ribosome, each subunit has a large ribosomal RNA and a small one. So 3 of the 4 ribosomal RNAs, 18S, 28S, and 5.8S. Anybody, what's that S mean? No. No. Let's put it this way. It refers to the size of the molecule. You've probably Svedberg units. No lights going on. Okay. I'm just too damn old. Okay. So basically, this is really old school that predates my entry into this business. And it was measuring the size of these molecules based on how fast they move through a sucrose gradient in a centrifuge. So the smaller the number, it's equivalent. It's pretty much equivalent to the size of the molecule. The smaller the number, the smaller the molecules. So the 18S is the large, this is in the small subunit, but it's the big ribosomal RNA. The 28S is the large ribosomal RNA in the large subunit. The 5.8S is the second ribosomal RNA in the large subunit. The 5S we'll get to in a minute is the 4th one. In eukaryotes, instead of scattering these genes all around, these 3 genes are clustered together into one unit, and then they're repeated tandemly, one after the other. So the cells, as we said, have to make a lot of ribosomes. They need to make a lot of this ribosomal RNA, and so even yeast cells, if I remember correctly, even yeast has about 250 copies of this 3 ribosomal RNAs. So the way this works, and don't confuse this with splicing. It's going to look like it, but it's not. They're organized together. So we have the dark sequences here are the actual coding sequences that are going to be in the mature ribosomal RNA when we're done. So this is the DNA sequence. Then we got the light region. So this is the sequence coding for 18S ribosomal RNA. This little one in the middle, 5.8S, and then the 28S. Now, this is transcribed by RNA polymerase I. So RNA polymerase I initially, just like with a pre-mRNA, makes a transcript that goes through all 3 genes. But then it goes through a processing, but it's not splicing, to remove this lighter colored sequence that's actually between the genes. So it's not splicing because we're not removing sequences within the same gene, but actually sequences between genes. So they make one large precursor and then clip away on the ends of the structural portions. The lighter colored sequences get thrown away, and you have the 3 ribosomal RNAs. Now, if you're interested in population genetics or you've looked at this at all, this, because I like to make things complicated, is this gray? Is that right? It's gray. It's lighter color region right in here was called the internal transcribed spacer region. So it's the internal sequence because it's between 2 of the genes. It's transcribed because it's part of the initial transcript that RNA polymerase I makes, and it's a spacer between the 2 genes that's going to get thrown away. This sequence can undergo changes in evolution, and it really isn't going to have any impact on the functionality here because it just gets thrown away. But because these structural genes are so highly conserved, it was pretty easy to be able to, especially using PCR, to really, and the idea is to look at the sequences between these genes and compare them between different species to see how much they've diverged away from each other or how similar they are. So a lot of early population genetic studies utilize this so-called ITS, or internal transcribed spacer region. That's not in your notes. I'm giving you that for free. Extra. Trying to make sure you get your money's worth. Okay. Well, the discovery of self-splicing RNA opened up sort of another area of debate, or maybe it answered a couple of questions, and that is, where did the first cells come from? So this idea that there was this primordial organic soup and somehow these molecules just spontaneously came together and created the first cell. Okay, how did it happen, kind of thing. But the finding that RNA can actually be has enzymatic activity suggested, and there's a lot of biochemical evidence that out of that organic soup, RNA was probably the first of the macromolecules. Between DNA, RNA, and protein was probably the first that would form spontaneously. But now you had a mechanism whereby the RNA could serve as the genetic material for being able to replicate the cell, replicate the genome, and it had enzymatic activity. Ultimately, the RNA could be replaced by the more stable DNA as a repository for genetic information, and proteins were more effective catalysts than the RNA, but now you have kind of a unifying idea that just RNA by itself could serve the functionality as genetic material, be able to replicate itself, and also serve enzymatic functions. Has anybody proven this? No. Okay? But it's a nice kind of intellectually appealing idea. Okay. Here's some of the crazy stuff. So you took general biology and they told you that there's tRNAs, ribosomal RNAs, and messenger RNA. And you were done. That was it. No, that's not true. Probably now, oh, okay. Yeah, go on. Has it been that long? Okay. Going back about almost 15 to 20 years. Okay. I have shoes that are older than that. Was the discovery of another level of gene regulation. So again, in general biology, they tell you, I'm sure that, correct me if I'm wrong, they probably say the cell makes the messenger RNA, it translates the protein, then the mRNA gets degraded. All right, well, that's not true in eukaryotic cells. Some messenger RNAs turn over very fast, others can last for days in the cell. And so you needed a mechanism of regulating gene regulation. Instead of just relying on the exonucleases to degrade the mRNAs whenever they got around to it, you wanted a way to target it and specifically regulate this. And so about 20 years ago, right around the turn of the century, was the discovery of some small RNAs that actually carried out that function. So there was 3 classes The siRNAs are small interfering RNAs, some microRNAs, just because they're just a class of very, very small RNA molecules, and the pi-Y interacting RNAs, which we're not going to talk about at all. The main thing I want you to see out of this, and again, another Nobel Prize comes out of this, and this was so important. Usually, the Nobel Prize just goes to really old people because you have to have some time to prove that your discovery was really important. This was so big that the discoverers got the Nobel Prize, I think, within like less than 5 years after they discovered it. And this is a process called RNA interference. And so it's a mechanism for gene regulation that's going to target a specific messenger RNA and degrade it, instead of just waiting for random events to happen. It also can be a protective mechanism against viruses and transposons. So it's a way for the cells to selectively target and knock out and destroy a particular messenger RNA. It starts, it's triggered by double-stranded RNA. So you can, just like you can have double-stranded DNA, you can have double-stranded RNA, except you just have to use instead of the T. So you got AU base pairing. How do you get double-stranded RNA? We have this thing called inverted repeats. Okay. So the cell can transcribe an RNA sequence that has inverted repeats, or maybe it has 2 different genes, but they make an RNA that's complementary to each other. So in one way or another, the cell's going to be making these double-stranded RNAs. All right. And then you got some rather interesting enzyme names here. So the first thing that happens is you have this double-stranded RNA and an enzyme called dicer. So I don't make this up. That's the enzyme. It cuts up the double-stranded RNA into little short pieces. Now, because they're very short and they're only being held together by the hydrogen bonding, they fall apart and you have lots of little short RNAs, single-stranded RNAs. They associate with some proteins to form a RISC complex, which is an RNA-induced silencing complex. What's going to happen is this RISC complex with this little short RNA is going to interact with its complementary sequence on its target messenger RNA, and when it interacts with that messenger RNA, it degrades it. If it doesn't degrade it, it blocks translation. So it's a way to knock out the expression of the gene at the level of the messenger RNA. It's still transcribing, it's still making pre-mRNA, it's making the mRNA, but it's preventing it from being translated. So this was a form of gene regulation, and it's now involved in apoptosis or programmed cell death, cell proliferation. But what probably got him the Nobel Prize was, let's say, cancer biologists working in tissue culture systems and such can't do the kind of genetics that, for instance, we can do with Drosophila. So they recognized right away, here was the mechanism. If I could get double-stranded RNA into the cells, I could do the equivalent of a genetic knockout, and I could interfere with the expression of my gene of interest. So it very quickly became this very powerful tool for using to study genetic mutations in cell culture systems where you couldn't do this kind of genetics. To show you how this works, we actually use this in the lab. We do this in Drosophila. In this case, it's a very elegant genetic system. It's about 30 years old, and I don't really have time to go into it. But in this case, what we have is a transgene that actually makes the RNA that can form the double-stranded RNA. That transgene is in one parent, but it's not active. It has to be activated by a protein called Gal4. We'll talk about this later on. And that's being made by the other parent. So this is some work that a former grad student did. And these are the 2 genes that we work with, MSRA and MSRB. And so this is our genetic, this is a Western blot. So it's been probed with an antibody we made against the protein. And so this first lane is our genetic mutant. So you can see there's no MSRA protein. This is the parent that makes the Gal4 protein, and it makes the MSRA. These are wild type for the 2 genes. This is the parent that has the transgene in it. It also makes the protein. But these are the progeny when we cross them together to activate the system, and now you see the protein's gone. Same way over here with MSRB. Here's our genetic mutant, doesn't make MSRB. Any guesses as to why there's 2 bands here instead of 1? Have you guys heard of this thing called variants or isoforms? Okay, that's what's happening here. Here's 2 of the 6 or so. Most of them are very close in size, so the gels can't separate them. But here, there's 2 isoforms. So these are both encoded by the MSRB gene. They're just spliced differentially by the cells. This is the strain. This is actually the same strain as this one. It also makes MSRB. This is the other transgenic line that makes the RNAi, and then you cross them together, and it's gone. Okay? So it's a really powerful tool. For instance, there's a laboratory called Genelia Farms Research. They're funded by the Howard Hughes Medical Institute, so they got a boatload of, they just back up trucks with money and keep dumping it in there all the time. And they're neurobiologists, and they want to work with, they consider Drosophila a tractable model system for neurobiology because the adult fly brain only has about 100,000 neurons. The larvae have about 10,000, as opposed to the millions, if not billions, of neurons that you guys supposedly have. Okay? What they're trying to do is set up this system where you can selectively knock out or turn on, however you want to set it up, your gene of interest in virtually every single cell throughout the entire nervous system of the fly, so that you can literally form bridges between the physiology of what's happening, the electron microscopy that shows you the morphology, and the biochemistry that's really going on as a real tool to be able to dissect this do neurobiology in this system at a level of resolution that you've never been able to see before. And so and the important point for us is they're making and constructing all of these lines and putting them on in a repository at Indiana University, and we literally can just go shopping for the ones that they want. I looked at it the other day. They have almost 6,000 of them up there already, and they expect to have this project completed over the next couple of years. So this RNA interference is really a major tool now at all levels of eukaryotic research. Okay, so this is the end of chapter 14, so it's probably a good stopping point.